So this is lab video number 2. I'm making this to cover 3 media: the starch plate, the gel tube, and Phenol Red broth. So the starch plate, I've got a plate here to show that, demonstrate that. I made a line on this side of the plate, and I made a line on the other side of the plate. I don't know how well you can see that. Bacteria didn't grow too well on this side, but you can see the bacillus grew well on that. And what I want to do now that they've grown on the plate, and if they've secreted amylase, they're going to break down the starch, and the starch is going to be stained with the iodine. So what I'm going to do, hopefully, is add some iodine to this plate so that it stains the starch. Okay, so the starch will appear dark. All this dark area is because the starch is still present, right? The zone that looks kind of yellow, orange color, that's there's an absence of starch there, so the iodine, or the indicator, doesn't stain that part of the plate. Okay, so the purpose of the starch plate is, do bacteria secrete amylase? Okay, the starch plate contains starch, of course. If bacteria have the ability to secrete the amylase, then they break down the starch, and you'll see a zone around the bacteria where the starch was broken down, right? You'll see this, what we call a clear zone. Now when you add the iodine, it takes it a few minutes to get into the starch, right? It has to get it, the agar, there's agar in this plate as well. So the starch just has to seep down into the agar itself, and then hopefully, I don't know if this is going to be as clear on this plate as it is on this plate. I kind of wanted to show you the plate and me adding the iodine to help you see what we're doing here. You know, this iodine gets added after the bacteria grow. So I made this plate last week. They grew up. The E. coli didn't grow well, and it looks like a couple fruit flies got in there, and they become larvae when they lay their eggs and become larvae, which are like little maggots. It looks like they went to town on they crawled around on the surface because they leave like little tracks where they've been. Yeah, I don't know if this is really gonna work all that well. It doesn't look like they secreted amylase, so I'm not really seeing much of a zone around the bacillus. And that happens occasionally. You can't force the bacteria to do anything. So if they make the amylase, you see a zone. If they don't make the amylase, you don't. So, but hopefully this is all clear, what I've written here. In your principle, right, you should talk about what I've got here. Starch is in the plate. If bacteria secrete amylase, starch is broken down. It's indicated when you add the iodine, and iodine stains the starch. So those are the kinds of things, right? This information belongs in your principle. It's hopefully pretty clear what the purpose is. So let's do the gelatin tube. Now I didn't inoculate this last week with the, if I had, I'd have taken a needle, and a needle is the same as this loop, right? But you have a loop on the end of it. Instead, you just have a straight wire, and I would inoculate, put bacteria on the wire, then I would stab the gelatin, and the gelatin in this tube turns there's no agar in it, right? So gelatin is just like agar. It's made of protein instead, but it turns it makes it a solid media. Okay? So if the bacteria secrete gelatinase, right, then they can break down the gelatin. That's the enzyme that breaks down gelatin. And instead of having a solid media, when you tilt it, if it were liquid, it would run down the tube, right? So there's no color for this positive test, right? If the test is positive, for gelatinase. We mean gelatin is hydrolyzed, that's what gelatinase does. Breaks it down into amino acids, you get a liquid. Now, if it's liquid, it can also turn to liquid if this room gets too warm, like around 80 degrees, it starts to appear liquid. The gelatin gets it stops being such a solid. So to confirm that it's not due to the temperature and it's due to the gelatinase, you put it on ice for 5 minutes. So you cool it back down. If it's solid, then it's negative, right? That's what a negative test is, solid. If it remains liquid after you cool it on ice, then it confirms the positive result. So you should describe the positive result in your principle, not just place on ice for confirmation. Tell me if it's still liquid after you place it on ice. So that's the gelatin tube. Then the last one for video 2 is called the Phenol Red broth. Now this one's a bit complicated. I inoculated a Phenol Red broth last week, and if you look on the top, it almost looks still red. It can be a mixed signal. Instead of becoming completely yellow, sometimes you'll see some red in it because it's going from yellow back to red. Well, it turns red because the pH goes down from the fermentation of whatever carbohydrates in it. So that's the purpose. Can bacteria ferment various? Now there isn't various carbohydrates, there's only one of them. But if there was in that tube, say lactose, then you're asking, can they ferment lactose? If we put sucrose in it, you'd be asking, can they ferment sucrose? You don't put both, you just use one. But you can, there's almost 30 different carbohydrates that you could use, right? There's plenty of them. Our lab just uses these two. So that's the purpose. The principle should contain the information about, right, the Phenol Red. It's called Phenol Red because that's the indicator in the media. And then you can put either sucrose or lactose or others, but for our purposes, we just use those two. So if those are fermented, you get yellow, right? And that's what this color looks like. Any yellow color is positive. Okay? The reason it looks cloudy is because there's lots of bacteria growing in there, right? These look real clear. The bacteria sometimes stay in the bottom. But they didn't ferment whatever. If it was sucrose in here, if they weren't able to ferment it, then it would stay red, right? The pH didn't change. So there's this what's called a Durham tube. If you can see that. A Durham tube is a test tube that's upside down inside the big tube. So if bacteria grow in here and make a gas, the gas goes to the top and it traps it. If it made gas outside the tube, you would never detect it, right? So it's used to trap the gas if it's produced from fermentation. You're not going to get any gas in a negative tube. Negative fermentation, no gas. So I don't know if you can see that very well. There's no gas in that tube. If you look, I don't know how well you can see the Phenol Red. The Durham tube is right there, and there you can even see bubbles. Right, this bacteria made a gas. So we know there's 3 outcomes. Right, if it's positive, you get yellow color, no gas. It can be positive with the gas. Okay, so there's 3 outcomes. Some bacteria don't ferment sucrose. Some bacteria do. Some bacteria ferment it with a gas. Then you need another tube to do lactose. If they didn't ferment lactose, it would stay red. If lactose was in here and they fermented, it would turn yellow. If they fermented lactose and made a gas in the process, then you would see a bubble in the Durham tube. Okay, so the purpose is pretty straightforward, get bacteria. And then this information should be included in your principle. Okay, so anyway, that's that video.